It is estimated that hypertension is present in over 100 million Americans. Hypertension contributes to conditions and diseases including strokes, loss of vision, heart attacks, and renal disease. The Renin-Angiotensin-Aldosterone system, or the RAAS, is responsible for the regulation of blood pressure through the release of hormones. This system is often overactivated in disease states such as diabetes and hypertension, with an elevated level of renin and prorenin circulating the bloodstream. Diabetes and hypertension are frequently comorbid and can both lead to chronic kidney disease. However, it is not well understood if diabetes or hypertension is the primary cause of CKD. Damage to the kidneys from both disease states is caused by chronic inflammation of the vasculature and glomeruli. Renal inflammation is mediated by pro-inflammatory cytokines secreted by infiltrating macrophages. These infiltrating macrophages promote inflammation through the secretion of cytokines in the kidney and heart, leading to diabetic kidney damage. It is important to determine the mechanisms at which renin and prorenin are released due to the elevated secretion from JG cells in models of diabetes. The current theory is that the enhanced reactive oxygen species and renin release found in models of diabetes is caused by specific cytokines secreted by infiltrating macrophages. Thus, identifying the expressed genes in infiltrating macrophages in a model of diabetes would reveal any genetic disparities with a control model that may promote renin release. The renal system consists of the kidneys, the ureters, and the urethra. The kidneys are responsible for the filtration of 200 liters of blood a day and the regulation of blood pressure. The overall function of the kidneys is to filter toxins, metabolic wastes, and excess ions. Filtration begins in the glomerular capsule, where fluids and solutes are forced into the bloodstream due to hydrostatic pressure. The rate at which filtration occurs is called the glomerular filtration rate. A normal filtration rate is determined by the membrane permeability, arterial pressure, and available surface area for filtration. Once the solutes have entered the glomerular capsule, they begin their journey along the nephron tubulars. The nephrons house a specialized structure that regulates blood pressure through the release of renin. This region is known as the juxtaglomerular apparatus and is formed by the glomerulus and the afferent arteriole. The Renin-Angiotensin system is responsible for the regulation of blood pressure through the release of hormones. It is composed of three primary components: renin, angiotensin II, and aldosterone. These three components work in concert to decrease salt delivery to the distal convoluted tubule and elevate arterial blood pressure in response to a decrease in renal blood pressure. Angiotensin II is a peptide hormone that acts on the brain, kidney, adrenal cortex, and arterioles by binding to the angiotensin II type 1 and type 2 receptors. AT receptors have been shown to cause vasodilation, meaning the widening of blood vessels, by stimulating the generation of nitric oxide. Angiotensin II's effects on arterioles result in potent vasoconstriction by binding to the G protein-coupled receptors. Renin is the single most important factor for blood pressure control since it is the rate-limiting step in the production of angiotensin II. Renin release is mostly regulated by three main factors: sodium tubular delivery sensed by the macula densa cells, adrenergic stimulation, and renal bearer receptors. Additionally, hydrogen peroxide, a reactive oxygen species, has been shown to be a potent stimulus for the release of renin from JG cells. Renin is expressed in renal juxtaglomerular cells as a propeptide. Renin splits angiotensinogen into angiotensin I, which is an inactive precursor to angiotensin II. The conversion between angiotensin I and angiotensin II is catalyzed by the angiotensin converting enzyme, or ACE, which is produced in the vascular endothelium of the lungs and kidney. This increases arterial pressure by transferring fluids into the blood volume and extracellular space. Inflammation is a natural biological response of the immune system to combat harmful stimuli. The inflammatory response is a complex coordinated network of many cells, including neutrophils, monocytes, and lymphocytes, which are all attracted to sites of injury. Macrophages are tissue effector cells that are derived from monocytes. They are important for inflammatory initiation and modulation. Macrophages present antigens, undergo phagocytosis, and produce cytokines and growth factors important to the regulation and maintenance of the immune response. Macrophages in injured tissues are not merely passive bystanders. They are present in significant numbers throughout all stages of kidney disease in the form of inflammation. Macrophages and monocytes are involved in the pathogenesis of hypertension. In the vasculature, macrophage-derived reactive oxygen species induce endothelial dysfunction, while pro-inflammatory cytokines induce epithelial dysfunction. This results in vascular oxidative stress and impairment of sodium excretion. Pro-inflammatory cytokines released by M1 macrophages include tumor necrosis factor alpha and interleukin-1 beta, which may cause an increase of blood pressure and lead to organ damage. Macrophages contain many phenotypes including M1 pro-inflammatory cells and M2 anti-inflammatory cells. A subset of M2 macrophages are strongly associated with renal fibrosis. Macrophages can differentiate into myofibroblasts, which have been linked to be a major contributor to renal fibrosis. The overall hypothesis is that the enhanced reactive oxygen species and renin release found in models of diabetes is caused by specific cytokines secreted by infiltrating macrophages. The specific goal for this project will be to identify the genes expressed in infiltrating macrophages in diabetes. The method includes 4 major steps: isolation of the infiltrating macrophages from C57 and Akita kidneys, RNA isolation, RNA sequencing, and data analysis. An Akita mouse strain is a monogenetic model for phenotypes associated with type 1 diabetes. The mutation of the insulin 2 gene causes the misfolding of the insulin protein. This leads to the toxicity of pancreatic beta cells, reduced cell mass, and reduced secretion of insulin beginning at 5 to 6 weeks. Akita mice on the C57 background contain high renal cortical hydrogen peroxide, high blood pressure, and become hypertensive at 12 to 14 weeks. A 12-week-old model will be used for the experiment to resemble high blood pressure in humans. The Kruger et al. Protocol was modified to isolate kidney-infiltrating macrophages. The kidneys from C57 and Akita mice will be perfused with oxygenated saline to ensure all circulating cells have been flushed. The kidneys will then be cooled and finely minced, then digested for 45 minutes. Intact infiltrating macrophages will be isolated using an immunomagnetic separation method using CD45 antibodies. For RNA isolation, a protocol will be adapted from Thermo Fisher's Trizol Plus RNA purification kit. Full coverage RNA sequencing will be performed using an ion torrent. This is done by using an ion total RNA sequencing kit, which provides a simple and effective workflow with as little as 100 nanograms of total RNA input. The Ion Chef system provides a streamlined automated library preparation, template preparation, and chip loader that will be used to prepare a template. The ion torrent will then sequence the genes of the infiltrating macrophages. RNA sequencing analysis is facilitated by Thermo Fisher's programs accompanied with ion torrent instruments. The data will be sent to the Bioinformatics and Biostatistics Department at Henry Ford Hospital, where it will be analyzed and sent back to the Hypertension Department. Further research of macrophages may lead to future treatment for diabetic-associated diseases such as atherosclerosis, nephropathy, neuropathy, and retinopathy. These diabetic complications are linked to the accumulation of macrophages in the affected tissue. Enhanced hydrogen peroxide is linked to an increase of renin and prorenin release, which can cause an overactivation of the Renin-Angiotensin-System found in hypertension and diabetes. Therefore, it may be possible to decrease the inflammatory response that contributes to hypertension in the kidneys by targeting genes in macrophages that promote the secretion of cytokines which enhance ROS production.